Good morning. My name is Shoaib and I'm here to present on the behalf of Philip Morris International. Today, my topic of presentation is toxicological assessment of the aerosol generated by a novel pin-based heating system in a human bronchial epithelial cells and organotypic small airway epithelial culture. The novel pin-based heating system is an electrically heating tobacco system that heats the tobacco to provide pulmonary delivery of a nicotine-containing aerosol. As shown on the left, the pin-based heating system is very similar to the tobacco heating system THS. The most important difference is that it uses an internal pin instead of a blade. The table on the right compares both systems. As you can see, PHS generates similar level of nicotine and aerosol delivery as THS. During the presentation, I will use PHS for pin-based heating system and THS for tobacco heating system. The aim of this study was to compare the biological impact of exposure to the aerosols generated by PHS and THS. The biological impact of this aerosol were also compared with that of cigarette smoke from 1R6F reference cigarettes. To this aim, we use monolayer of normal human bronchial epithelial cells to perform real-time cell analysis and high-content screening in what we call the 2D part of the study. Additionally, 3D organotypic culture of small airway epithelial cells were used to assess the impact on gene expression, histology, cilia repeating frequency, barrier integrity, cytotoxicity, and secreted factors. For the 2D part result, real-time cell analysis. In the real-time cell analysis, the greater the electrode impedance, the greater the number of cells on the bottom of the well and hence greater cell viability. For cigarette smoke, we found a dose-dependent reduction in the cell viability with a half maximal dose concentration of 0.045. On the contrary, there were no significant changes for PHS or THS aerosols. For the high content screening, the central figure shows the result of the high content screening. In this figure, the bigger the area, the bigger the effect, meaning that a lower concentration is needed to elicit this effect. With regards to the cigarette smoke has a similar impact at 4 hours and 24 hours. And decreased mitochondrial health membrane potential was one of the strongest effects, suggesting that it may be one of the first adverse effects occurring upon the exposure. Those effects have probably contributed to the observed increase of oxidative stress. Not surprisingly, we also found cell stress indicated by the increased translocation of season stress kinase along with the sign of DNA damage. With regard to the THS, we found a reduced impact on all analyzed endpoints when compared to cigarette smoke. Nonetheless, as with cigarette smoke was observed, a decreased mitochondrial health and increased oxidative stress with ROS and glutathione. No evidence of genotoxicity or apoptosis was found. For PHS aerosol in general, the activated endpoint and corresponding minimal effectiveness concentration in PHS matched those of THS. For the histology part, the tissue were exposed, and after 48 hours of post-exposure, histology tissue was stained with three different types of stain: hematoxylin, iocine, and Alcian blue. The figure on the right shows a representative picture of three different cultures for each treatment. Culture exposed to THS or PHS aerosols had a morphology similar to the culture that were exposed to air for all puff tested. Culture exposed to cigarette smoke for more than two puffs had pronounced tissue damage and thinner epithelial culture. Cytotoxicity and barrier integrity was also measured. To assess cytotoxicity, we measured the activity of adenylate kinase in the basolateral media. The more kinase activity observed, the more leakage, and therefore the more cellular damage. Exposure to THS or PHS aerosol did not impact cytotoxicity. For the barrier integrity, tear measurement was performed. Epithelial barrier integrity was assessed, measuring the transepithelial electrical resistance or tear at 48 hours after the exposure. In contrast to cigarette smoke, tear values in the presence of PHS and THS aerosol were comparable to corresponding air-exposed culture. Ciliary beating frequency. Regarding ciliary beating, the upper graph shows the ciliary beating active area while the lower graph shows ciliary beating frequency. All culture had a similar baseline beating frequency and the active area. After the exposure, 48 hours in general, cigarette smoke led to a reduction in the active area and the beating frequency. THS aerosol and PHS aerosol presented similar values for active area and the frequency. Those values were in general higher than in culture exposed to cigarette smoke. Concentration of secreted inflammatory mediator were assessed in basolateral media of the culture at 48 hours of exposure. As you can see in the figure, the concentration of several factors IL-1 alpha, IL-8 and MMP-1 increased with the puff number of the cigarette smoke. Exposure to the THS or PHS aerosols did not result in a dose dependent alternation of any of the assets inflammatory mediators. Gene expression, a causal network enrichment was conducted based on the gene expression profile of culture exposed to cigarette smoke, THS, and PHS aerosols relatively to air for 4 hours and 48 hours post-exposure timing. The Nectar model were used was cell fate, cell proliferation, cell stress, and inflammatory process network. The graph on the left shows the biological impact factor BIF, which reflects overall impact on all Nectar models. The highest BIF was observed with cigarette smoke exposure and defined as 100%, and this was the reference. In general, exposure to PHS or THS aerosol for any puff number resulted in lower BIF values and MPS scores than exposure to 1R6F cigarette smoke. Relative change comparison impact of THS versus PHS. The overreaching objective of this study was to determine whether the reduced impact of PHS aerosol was comparable and not worse that the reduced impact of THS aerosol, both relative to cigarette smoke. This was considered fulfilled if the reduction of impact for PHS was at least 80% the reduction of the impact for THS. Result for cytotoxicity, impact of 8 and 10 puffs of cigarette smoke on cytotoxicity was expressed as 100%. The main reduction in cytotoxicity of PHS aerosol versus cigarette smoke were more than 80% of the mean reduction in cytotoxicity of THS. The relative change comparison impact of THS aerosol versus PHS aerosol for the tear and also for ciliary beating frequency and also for different secreted mediator were also measured. Value of 10 puffs of cigarette smoke were expressed as 1. The tear value associated with 10 puffs of PHS and THS aerosol were computed relative to this cigarette smoke reference group. Again, the impact of PHS was comparable to the impact of THS for same puff number. A similar results were obtained for ciliary beating values and secreted mediator for THS and PHS aerosols. The biggest difference was appeared for CXCL10, IP10, a pro-inflammatory cytokine triggered by IFN, which were increased around 70% relative to cigarette smoke for PHS and around 90% relative to cigarette smoke for THS. This was not considered clinically relevant. For the conclusion for this study is like PHS and THS aerosol induced comparable cytotoxic and phenotypic effects which are markedly lower than the effect induced by the cigarette smoke, 1R6F, in human bronchial epithelial cells. PHS and THS aerosols up to 50 puffs trigger fewer biological effect than cigarette smoke, 1R6F, up to 10 puff only in human 3D organotypic small airway epithelial cultures. Relative change comparison allow us to conclude that the impact of PHS aerosol on organotypic small airway epithelial culture is comparable to and not worse than the impact of THS aerosol. Thank you very much for listening this presentation.